Okay, I hope my slides are visible now. Okay, you can review the lecture objectives when you get a chance. And when we look into research strategy is the description of the rationale of your research and how you plan your experiments to accomplish your objectives. As we discussed in our specific aim, we may be having 2 to 3 specific objectives or specific aims. So in research strategy, we need to give a clear-cut description of how and why we are going to accomplish those aims following the certain set of experiments based on our expertise. Okay. So this research strategy can be divided into 3 major sessions. That's where the reviewers look into. These are the review criteria. Basically, 3 major sections can be composing with our research strategy. First of all, the significance. Significant significance page should emphasize why this research is very important, how it is significant in the present scenario. Second one is innovation. Innovation need to emphasize how the proposal is novel and unique, how our proposal is different from the ongoing studies or already completed studies. And the third one is our approach and how and in what all strategies we adopt to solve the problem or to answer our research questions. And we need to have strong preliminary data to support all these 3 components, and definitely it has to align with our specific aims. In order to define research strategy, I would say research strategy is the map that shows the reviewers how you plan or how you organize the entire research to test your hypothesis and attain your objectives. Okay, this is a roadmap, and basically, as I mentioned, as I repeatedly emphasized during our last session, always keep this thing in mind that the reviewers are going to be the primary people who are dealing with this application. So always think in a reviewer's perspective when you write your research strategies and your research strategy and other sessions. Okay. In your research strategy, you need to lay out the experiments as well as the anticipated outcome. Our primary goal is to convince our reviewer how likely our research is going to be successful and give a very broader overview of that. And always ask these questions when you articulate your research strategy. Before you put each and every statement, ask this. Can this research move the field forward? Okay, then is this field important? That means whether our approach is going to make any difference in the field. So whether we can move the field further forward and whether it can make any difference. And research strategy can be organized into multiple ways. The commonly followed approaches are option 1. You can simply start with the significance, innovation, and approach. And then you can give your preliminary studies in your approach, and you can describe aim 1, aim 2, and aim 3. Or you can go with your aim 1. You can split significance, approach, and innovation in your based on your aims. Okay. The third one would be, okay, you can mix everything together with preliminary study. So depending upon how you want to organize, how better you can organize, you can choose any of these options. However, emphasize that, make sure that you have all the major components included in your application. And research strategy, why it is important? Because research strategy critically review the research plan through the lens of a reviewer. So when you articulate, always see through the lens of a reviewer to identify the potential questions and weak spots. Okay, when you write an application, if you know there is a weak spot, you yourself should be able to identify and acknowledge that there, so that the reviewer should be aware that, okay, you know what is going to be the weakness and you have alternative plans accordingly to address those issues. Okay. And always emphasize on the experience which the team has. Okay, for example, as I mentioned during our last session, for example, in your team, if you are doing with some high throughput or highly advanced techniques, including single cell genomics, and if you are not well experienced in that, you can recruit someone who is having significant experience in that particular technology as a consultant or as a collaborator or as a co-investigator, and mention wherever you mention single cell sequencing, you can mention, okay, these experiments are directed by or will be conducted under the supervision of so and so people. Okay, and also just mention if you have any specific instruments required, if you have those things, or if you have access for those things in your within your collaborators, you can just very well make that. Always seek support from your own preliminary data. That is very, very important. So we need to have foundational data. We need to show that it is doable within our perspective or within our environment, so that the reviewers will be confident on your data as well as on your research. Okay. And ensure that the reviewer will be having fundamental understanding regarding the field, and he knows how to fit your work into that particular field. So keep that in mind. So reviewer will be having a fundamental knowledge, not may not be having a deep in-depth knowledge, but he or she will be having a fundamental knowledge that can be fit into any research discipline or approach. And also another important thing, preliminary data. Preliminary data is very important to demonstrate the competency of methods, interpretation, as well as to provide a strong scientific rationale for the proposed experiments. So if you have strong preliminary data, if you are proposing an experiment, if you have a feasibility data that can be incorporated in your grant application, that will give an impression to the reviewer that yes, these people, they have experience doing it and they know how to handle these type of experiments. Okay, when you do those preliminary data, give very accurate information. Don't give any generic thing. Make sure, emphasize that, okay, this is going to work 100%, and give each and every details, fundamental details. No need to give the detailed methodology or so like that. And if you have already published something related to that, you can very well cite your own articles there. And there could be other sessions where you can again incorporate those information, including animal, subject, vertebrate animal, select agents, and these may not be counting into the research strategy page limit, but you can mention that, okay, additional information can be found in these sessions too. And also always bring the expertise, cite your biosketch. We need to give our biosketch or biodata. There you need to cite what our expertise are. And in the research strategy also, okay, he or she has particular experience in running these type of experiments. And when we look into the significance, provide a clear focus on the current understanding regarding the basic biology, importance of research in that particular field, identify the knowledge gap, give new research opportunities and new directions and findings. Okay, for that, you need to clearly describe the prior research as well as your own preliminary studies to provide a scientific rationale. And also emphasize the strengths, weakness, and the pitfalls of the studies which you are proposing and how you are going to address those. And one major mistake which we all make as grant writers is the redundancy. We will be keeping on repeating same things again and again. That will kill the enthusiasm of the reviewer. So give try to emphasize on minimal redundancy. But if you have a key concept, you may be able to repeat a little bit. But don't repeat the methodology or don't repeat the same fact again and again. That may kill the enthusiasm of the reviewer. And always give preliminary data. Definitely, it is required for the significance. And give long-term goals or long-term achievements which you are planning to execute through these grants. Okay. And then additional tips, give the importance of your hypothesis in that particular field or human disease, and point out the significance throughout the application. So whenever you make any comments, always emphasize why that particular aspect is important in this field. Okay, you can point out the opportunities, gap in the knowledge, the research which needs to be done, and the roadblock, what are the ongoing research, and also try to emphasize how your project will fit into or address any of these knowledge gaps. So in and also you need to give a clear cut picture how the mission of funding agency to improve science will fit into your application. So give that aspect also when you write your significance page. And in general, significance can be organized into three or four paragraph. If I go, I will go with four paragraphs. For in the first paragraph, I will introduce the problem, the ultimate goal, and the already ongoing or already completed research from the literature, and why this addressing this problem could be very useful if you consider the advancement in science or the patient care. So I will give a broader umbrella there in the first paragraph. In the second paragraph, I will be giving the most important points supporting the introduction, providing additional background. It will be basically narrowing down to our aspects. In the forthcoming or third paragraph, I will little bit give an overview of the approach and difficulty, and I would emphasize the gap in the knowledge and how our approach is going to address mend those gap in the knowledge. And I will be explaining and highlighting how potential barriers may arise and what are the alternative approaches to be proposed in this research, and how other people, when they encountered such type of pitfalls, how they overcome those pitfalls. So all those things will be giving, and I will be giving some insights into the experimental methods, experimental approach, and new strategies, which is uh, describing in the research strategy session. And in the final or fourth paragraph, I will again articulate the significance in a broader context. I will make the storyline in such a way that will end up in bringing out our hypothesis and how the proposed research is going to support or to enhance or improve the ongoing or the advancement in the knowledge in the field. And also, I will be giving the feasibility of this research by providing the timeline, by providing the expertise of the collaborators, as well as with the preliminary data. Next important avenue to look is innovation. Innovation means you need to include sufficient evidence based on your preliminary data to convince the reviewers that you are rationally strong, you are supporting data, your preliminary data is supporting your approach, and your study is clearly feasible. At the same time, it is conceptually, technically, and technologically unique and innovative. So demonstrate how our proposed research is unique and new compared with the ongoing or published research literature. Explore new scientific avenues which nobody has ever tried. Come up with a new or novel hypothesis. Give emphasize that, okay, if we address these hypotheses or if we test these hypotheses, that will create new knowledge, and you can always keep the room for refinement, improvement, and propose new application for an existing concept or a method. For example, if somebody developed a approach or a strategy for cancer related diseases, you can adopt that approach for cardiovascular disease, for example. So that means just identify a new application for existing innovation. So that will also help the reviewers to understand the innovation better. Always make the reviewers feel confident that, so the risk in this proposal, even though it is new, nobody has done, it is worthy taking that risk. So that emphasize that thought process you need to generate when you write innovation. And every time the mistakes which we made, we will be putting too much innovative aspects that will kill again the enthusiasm of the reviewer. And we need to always show there is a paradigm shift in the area of science, so that reviewers can challenge the feasibility and the outcome and give a strong case for challenging the existing paradigm. You need to clearly mention that, okay, these are the existing challenges. That's why we are taking this route to address those. This will be an innovative approach so that it can be adopted by other people, other scientists from other disciplines as well. Definitely, these innovative aspects should be supported by our preliminary data. And next, when we look into the approach, always think that the expected reviewers, the assigned reviewers, are expected to scrutinize the approach. That means they will want to know what you plan to do and how you plan to do. If you remember the two potential questions which I asked during our last session, should it be done? Can it be done? Should it be done means whether it is significant or worthy to do. Can it be done? Whether it is feasible. Okay, so we need to have rigorous study design to understand, to address the problem which we identified, to prove the hypothesis and sub-hypothesis, and to achieve our specific aims successfully without any bias or without any hurdles. So the methods for blinding, if we are dealing with some experiments which deals with human patients, for example. So we need to be blind. We need to have a blinded approach in generating data as well as data analysis, which needs to be clearly articulated in the approach session, and how we go with the treatment randomization, and how we address the biological variables, including gender, or geographical variabilities like that. And also, when we organize the approach session, and start with a bolder heading to emphasize or to articulate the specific aims. Under each aim, we can have sub-aims where we can describe the experiments and also outline the branching for the next step, how we are going to proceed to the next step. For example, if you get a result X, you will follow the pathway X. If you don't get result Y, you will follow pathway Y. Like that, you can give, okay, if I get this, I will go this. If I don't get, I will go that route. It will be worthy considering those experimental approach in a self-explanatory flowchart. Another approach, another thing to remember is spell out at least few sets of experiments. If they are unique or if you are very specific or that particular experiments, just emphasize that. And it is always a good idea to reinstate the key points regarding the significance and impact uh, when you put your approach. You can again rewrite your specific aims. Okay, this is what we are going to do. And if you published your own articles, just cite those that will give confidence to the reviewer that you are able to conduct those experiments. And if you are designing some new experiments, give clear picture, clear idea that how you are going to do, what approach you are going to take, and how you will be successful, or how do you think or why do you think this is going to be successful. Those information you need to give over there. And in approach, keep these things very carefully, consider these things very carefully, omit all unwanted information. If you try to over impress the reviewers with your knowledge, definitely there will be rooms for finding flaws. And also, don't give the reviewers any opportunity by including which you don't need. So there will be a tendency to add all that what we need. We have no need. Just give only minimum essential information. Give only essential information to drive your project. And the common mistakes which people make when writing the approach, every time they will be overly ambitious. Overly ambitious means they will pack with too much experimental details and too much data, considering that which may not be feasible or which may not be completed within the time frame, and based on the qualification or experience of the collaborators. And also the aims could be underdeveloped or unfocused, again, without any alternative approach or backup plans. Those things reviewers may not like. Make sure all your experiments are strategic. That means if you have something in your lab, it doesn't mean that if you have a particular instrument in your lab, it doesn't mean that, okay, since you have that instrument, I need to put that in the grant. No. Whatever experiment which you are planning, it should be clearly aligning with your proposed research and your specific aims to test your hypothesis. If the aims are too risky, for example, if you are generating some animal models where you feel like only 1% of the animals are going to survive or 10% are survive, that means that is a very risky proposal. So avoid such type of emphasis in the grant. Okay, you need to find some alternative ways. If you find, okay, this proposal is, if this particular model is risky, you need to develop some lower risky approaches to address those things. So if there is more risk, there is less likely for funding. And little description or interpretation of result and analysis. For example, if you are putting preliminary data, you are the authority for your research. You know everything. It doesn't mean that the reviewer who is going to read your data is going to understand everything. So if he is not able to understand, he will table your application. So give clear interpretation of result. Okay, when I treated my cells with compound X, I got the upregulation of gene Y. What does it mean? That information should be there. Okay. And don't try to rely too much on preferred hypothesis. Then they will think, okay, this is more speculative grant. We need to always have support from our preliminary data as well as from the published research. Okay. And some common tips when you write information. Ask these questions. Why are we doing this experiment? Is it relevant or not? Is it significant or not? Then what critical information will all this experiment provide? Okay, do I going to get anything novel? Okay, what will it mean when you get data from the experiment? Okay, if I get some data, interpret it. How I am going to use it for the advancement of science. And what are the anticipated results? If if we don't get any anticipated results, what will you do? Okay, what is your approach when your experiment doesn't work? And also, you are getting data, and how will you confirm the data which you get from each experiment is reliable? And also, did you try these experiments before? Do you have experience working with these experiments before? And also, how likely is your plan is going to work? So ask these type of questions. These are the questions which a reviewer asks on a common sense. Ask these questions, try to address these questions whenever you write anything on your approach level. And also give a subsessions for each aims regarding the expected outcome. You need to mention quantifiable and measurable outcomes to show the success of the research. And also always include some outcome metric. Okay. For example, if I treat the compound, I am expecting if I get gene expression maybe higher than twofold, I can make it as a successful thing. So likewise, you just make some successful success metric. How do you determine the success? So those things are important. And also clearly define the outcome criteria. You need to clearly define the endpoints. Okay. For example, upregulation or inhibition of genes, or functional recovery of organs, or the complete wound healing, like that. Depending upon your hypothesis, you need to clearly emphasize the outcome metric. Okay. And also be ready with alternative strategies, which should outline what to do if your primary approach fails. These are proposals, right? Whenever we do actual experiments, then only we will understand whether this is going to be successful, or is there any weakness emerging or so. We don't know. So give an alternative strategy, one or two alternative strategies to address if your primary approach fail, including the contingency plans for each specific aims and the expected challenges. Okay, so that will give an impression to the reviewer that, okay, they know the possible pitfalls and they know how to tackle this. Okay, so show reviewers how you plan for spending your grant period, for example, 4 years or 5 years or 3 years when you get funded. Okay. And also include multiple risk aspects as well as alternative. Always be aware and consider the limitations, potential limitations of the studies, and come up with alternative interpretations. Okay. And point out any procedures, situations, or materials that can cause potential hazard to the personnel or the animal or the investigators, and always give clear picture or clear explanation for how and what precautions are to be exercised. So these can be included in the alternative strategies. And also additional components include competency of the research team. If you have 2 or 3 co-investigators and collaborators, give what are their potential strengths and how their strengths are being harvested for our experiments and successfully completing our approach. Always give statistical analysis, how you are going to statistically convince your data. And if you are doing with any patient related studies or animal studies, always give power analysis to show how you identify or how you formulated the experimental groups and study population. Give a timeline, overall impact, as well as future research. Okay, what is your end goal? Maybe you need to bring your next 15-year, 20-year goals based on the data or based on the outcome from this proposed research. So these are additional components which needs to be included that can excite the reviewer. And that's all about writing the research strategy. In this PPT, I gave some applications which are funded through NIH from the NIH website. You can look into these applications and see how they articulated, how and why their grants were successful. Okay, that's all about the second session of research strategy. And if you have any questions, feel free to ask now. Any questions? Okay, we have an assignment. You need to write a research strategy, not more than 6 pages. It should be 6 page. Don't go beyond 6 page, anywhere between 5 to 6 pages. And you are already start writing the specific page. I think the deadline is on 15th. So when do you think you can provide this one along with the specific aims?
Hi, Dr. Thankam.
Yes, Heba.
Um, Perhaps maybe a little bit more time, just because 5, 6 is also the research strategy. Is it for the specific aims you said we could use our own
Oh, yeah,
current?
yeah. So research strategy and everything. You use your own research. So
Okay.
that will give you more in-depth knowledge regarding your research,
Hmm.
and also that will be helpful, right, for your ongoing research. So go with
Yes.
that.
Okay,
Yep.
thank you.
What do you think about 20th?
I think that's fine. So another 5 days.
Yeah,
Thank
yeah.
you. Um,
Yeah, send everything together to Natalie. I will get it from Natalie, and then we will be having a session to uh, discuss our applications. I mean, how you prepare and those things. I can give some insights.
Thank you, Dr. Thankam.
No problem. Okay, any further questions? No, hearing none, we agreed that those assignments, including 1 page specific aim plus maybe 6 page, maximum 5 to 6 page research strategy based on your own summer project, and you will be submitting to Natalie by 20th of July. Yeah. And maybe I think we have another session on 27th or 28th where we will discuss uh, the applications, and we can further discuss if any room for improvement, or I can give my comments and remarks. Yeah. Okay. Sounds good. Thank you. Any further questions? Yeah, if you have any doubts or if you need any support,